Hi, my name is Sakshi and this is my STEM Fair project. So my question that I'm trying to answer is how genetic mutations affect the heart. So here, my hypothesis was that if a mutation occurs in a heart-related gene such as MYH7, then the heart's normal rhythm will be disrupted. And in order to test this, I can't directly test the heart, so that's why I use gelatin. Gelatin symbolizes the heart and kind of stimulates how the heart works, the heart's muscle texture works. That's why I use the heart. So in order to test this, I used 10 cups. I labeled all my 10 cups, and then I poured 5 cups of water into the boil and I heated it. And after heating it, I took out 5 teaspoons of the Knox gelatin powder and I mixed it all in. And then I placed half of them into the 5 normal cups and I left them aside. And for my mutated, I need to have a constant and a difference, right? So for my mutated one, I added a half teaspoon of salt to the remaining gelatin. I mixed it all up for a minute or two, and then I put it in the refrigerator, both for 4 and 5 hours. And then once I took them out, I had to do my actual test, which is the part B of my test. For part B, I placed a quarter on top and I measured the dent depth. So for example, for my first one, my dent depth was 2 mm, and when I took it out, the seconds it took to recover was 2.5 mm. I did the same thing, and this is my a graph that I have, and this is my table. This is my average table, which shows the significant difference between the normal and mutated. This is my graph, and my conclusion. For my conclusion, the hypothesis was indeed supported because the stimulation in this model caused many changes in the structure, suggesting that genetic alterations can weaken the strength of the heart. So here's my DNA model. This is my normal model, and this is my mutated model. This normal model shows that everything works out well. However, my mutated model has the guanine and the thymine kind of switched up. So this is my project.
So our project was downforce versus drag, the physics of F1 cars. So our hypothesis was basically that if the rear angle of an F1 car is increased, it would go faster. And our procedure was basically we lined up 3 meter sticks or so, and then we put one in the finish line and one sticky note for in the finish line and one that follows. So, and our data shows that as the angle increased, the faster the car got. So like in the 185, it was the fastest angle we got by far.
And we observed that the higher the angle would be, the faster we would go. As you can see in the average, there's 3 trials for each angle. And as you see, like the 130-degree angle is by far the fastest than like any of the other angles. And every these sticky notes is like a meter, which we would always start on the first, and then we would end all the way on the third.
Great, back to you. All right.
And in conclusion, we realized that.
Can you do it one more time?
Yeah.
Okay.
In conclusion, we realized that as the car's rear angle is increased, the faster the car would go, since more downforce is interacted, and and less the angle, the less the downforce would be increased. So basically, drag.
Hi, my name is Brianna. This is my topic: how much electromagnetic radiation is emitted during different types of cell phone use. My hypothesis was if cell phones emit a measurable amount of radiation during different types of cell phone use, and the amount of radiation increases, then it will be the highest during a cell phone call. My independent variable was the type of cell phone being powered on, and then my dependent variable was the amount of electromagnetic radiation emitted by the cell phone. My materials that I used for this experiment were my measuring tape, my cell phone, my data table, and a volunteer with the phone. Also, this meter. My procedure was to set up in an area that was far from Wi-Fi routes and collaborate the EMF meter, measure background radiation, and put the phone in idle mode, and put on the call, the streaming, and the texting and charging. In my observation, I noticed that when calling, it's at 168 volts per meter, texting, which is at 162 volts per meter, streaming, 156 volts per meter, and charging, which is the highest, at 736 volts per meter. My research showed that electromagnetic radiation can be found during cell phone, Wi-Fi, TVs, radars, X-ray machines, tanning beds, suns, stars, atmosphere, and cosmic sources. When measuring radiation, it is absorbed by body tissue, and the cell phones emit radio frequency radiation. My conclusion is that when charging your phone, it is the highest, and you shouldn't do something like that. Also, my hypothesis was not supported because I can see when calling it's at 168 volts per meter and charging is at 736 volts per meter.
Hi, my name is Harshita, and this is like our project, and it's about hidden pigments of the flowers in flower chromatography. And our hypothesis was that roses, Peruvian lilies, and carnations all have different hues in flowers because they look more orangey or pinkish. So, and then our variables were that independent variables were types of flowers, and dependent variables were pigment separation and distance traveled by pigments. And our procedure was that we picked 3 different types of flowers that were red, and we cut filters for like coffee filters, and this is the amount of pigment we put on each of them. And we marked a pencil line for 1 centimeter from the bottom of each chromatography strip. To create the solvent, we used 75 milliliters of 90% isopropyl alcohol with 30 milliliters of water, distilled water, in a clean jar, and we poured the small amount of it in another jar to like put the chromatography strips in to test our hypothesis. And these are the flowers we tested. This is the research.
So our conclusion was that the roses, Peruvian lilies, and carnations all had separate pigments. So we used the retention factor to see if they have the same pigments. All of them had relatively the same retention factor, but their color separation was very different. The roses had more of a purple, and the Peruvian lilies had like a faded red, and the carnations had a deep pink. The pigments had to be the same. If the pigment separation has to be the same for the pigments found in the flowers to be the same. The reason why they're different is because all of them have the same pigment group, but inside the pigment group there are different types, and all of the flowers have different types of pigments inside the group.
Hi, my name is Aisha, and this is my project, Light That Shatters Life Itself. My question was, how does UV exposure affect DNA recovery? So DNA stands for deoxyribonucleic acid, and is the molecule that contains the genetic information organisms need to develop and grow. DNA is made of 4 base pairs: adenine, cytosine, thymine, and guanine, ACTG. And over here is a DNA model of the 4 base pairs. DNA is tightly packed into chromosomes. We have about 3 billion base pairs, and these are all in chromosomes. We have 43 chromosomes in 23 pairs. DNA can be damaged by environmental factors like heat and moisture. It can also be damaged by mutations, which are changes in a DNA sequence. There are a couple types of mutations, but there are 2 main types: somatic mutations and germline mutations. Somatic mutations happen inside your body, while germline mutations are inherited from your parents. There are a couple types of mutations in these categories, such as SNPs, or single nucleotide polymorphisms, silent mutations, and chromosomal mutations. Chromosomal mutations have many different subtypes. I listed 4 of them: inversion, deletion, duplication, and translocation. Inversion is when a DNA segment deattaches, flips, and reattaches to the chromosome. Deletion is when a DNA segment is lost, and duplication is when a DNA segment is copied. Translocation is when a DNA segment breaks off and fuses to another chromosome. An example of this is when chromosome 9, a segment of it deattaches from its chromosome and fuses onto chromosome 22, causing chronic myelogenous leukemia, which is a slow cancer that is related to like the overproduction of white blood cells. We as humans receive alleles, and we receive 2 alleles, one from each parent. We have dominant and recessive genes. Dominant means you need only one gene to appear, and recessive means you need 2. The electromagnetic spectrum comes from the sun. It includes radio waves, microwaves, infrared light, visible light, UV light, X-rays, and gamma rays. UV is subdivided into 3 categories: UVA, UVB, and UVC. UVA is found in nail lamps and tanning beds. That is a UV light right there, UVA. UVB is commonly used to treat medical conditions such as psoriasis, eczema, and vitiligo. And UVC is the most dangerous type of UV, but the Earth's atmosphere blocks it, so it cannot reach us. I use strawberries for my experiment for 2 reasons. 1, I can't use human DNA. 2, strawberries are octoploid, meaning they have 8 copies of their typical chromosomes, meaning they have a lot of DNA to be extracted. For my hypothesis, I said that if ultraviolet radiation is put on DNA, then the mass and visible amount of DNA will be lesser than when not exposed. To test this, I created an experiment where I had 3 beakers. I had 2 same-sized strawberries in each beaker. I mashed the strawberries and mixed water, salt, and dishwashing detergent, and I put isopropyl alcohol in them so that the DNA would form. Once the DNA formed, I put one under that UV light, one on the counter, and one in the dark. After 10 minutes of each exposure, I took them and took out all the DNA and extracted all the liquid, and I massed it on a weighing scale. These are the masses from each trial and each category, and these are the average. This is the average mass from each trial. And I made graphs here. The UV exposed one had less DNA than the dark one, and even in these, the shade had the most in most of them. And under a microscope, I observed that the UV exposed one had shorter and more fragmented DNA, while the shade and dark ones had much smoother and finer fragments of DNA. In my conclusion, my hypothesis was supported because, as you can see here, the UV exposed DNA had the lesser mass and visible amount of DNA than the other 2 categories. This is because when DNA is exposed to UV, the adjacent thymine dimers collide with each other and covalently bond, causing a twist in the DNA helix, and eventually leading to the disruption of the cell replication, which ultimately leads to the DNA being more fragmented, and it can even be dissolved. So that is the reason why DNA gets damaged under UV light.
Hi, my name is Deshant, and this is Eason over here. Yeah, so we're doing what operating system is the best for older computers. So, Eason, do you want to lead off with our research?
What operating system is the best for older computers? Our research is an operating system is a software that manages computers' hardware and applications, send resources, including memory, CPU, input and output devices, and file storage, and etc.
Right. So Linux distros are the most popularly used operating systems in the world, as they're used in phones that are based off Android, while Windows is the most popularly used operating system for computers. Our hypothesis and variable claim that both Ubuntu and Debian, which are both Linux-based operating systems, are going to work better than Windows, right? So the materials we really use are a 128 gigabyte SSD that you can get off Amazon for $30, and PTM 7950, 91% isopropyl alcohol, SATA to USB connector, and then the 3 laptops we used to test right over here. So we have a procedure which is disassembling the laptop, cleaning out the thermal paste on the CPU by taking out the fans, cleaning the fans, reassembling the laptop, installing the new SSDs. And when you close the laptop back up, you can plug in a USB drive with the desired operating system you have by getting Rufus on a working computer and then the ISO file for the operating system you want. And then from there, Rufus will take care of the rest. It'll tell you how to format the drive to get the operating system working. So over here, we have our observations and data, which which is where basically our conclusion is. We found that Ubuntu seems to be the least demanding, while Debian and Windows are more common than Ubuntu. Debian looks the best, but isn't as demanding as Windows. And Windows is the most commonly used one. Windows was performed 3 times worse than Debian or Ubuntu combined. So we believe that most people should switch to Ubuntu or Debian when they have an old computer. So over here we have our conclusion. In conclusion, our hypothesis was proven correct, and when we stated that Ubuntu and Debian are going to work better than Windows, we were correct. Ubuntu and Debian performed way better than Windows, with Windows using up at least 70% of the system while standing idle. So we have our graphs over here that show that our evidence. It shows that Windows actually performed worse. Thank you.